So today we're going to do some further reading on a paper that just came out in January 2026 around immune dysregulation in severe COVID-19. It's a pretty cool paper, but I'm not going to waste your time. It's going to be of most interest if you're a student that is studying topics like infection and immunity, single-cell transcriptomics, sports and exercise medicine, or pathophysiology and clinical science. If you're a researcher, it's going to be of most interest if your project is something around maybe severe respiratory infections, monocyte or T-cell dynamics, IL-6 JAK STAT driven pathology, or vaccine immunomodulation. That's not to say, of course, that this paper isn't going to be interesting to a wider pool of students and researchers than that. We don't gatekeep here, so if you're outside these topics, still grab a cuppa, come on in, and let's do some further reading together. So in this study, the researchers set out to understand why some people develop severe or critical respiratory infection, while others recover with much milder disease. They analysed almost 300,000 single immune cells from patients with moderate, severe, and critical COVID-19, as well as from individuals who had recovered. They also examined how the immune system responded after receiving low or high dose of this WSK V102C, which is a trivalent recombinant protein vaccine, which, if you've not heard of it, I'll give you a quick overview of later, don't worry. In terms of results here, what they found was that across the immune landscape, severe disease was characterized by a major shift in the balance between innate and adaptive immunity. One of the strongest signals in the data was the expansion of CD4 positive monocytes, which became highly inflamed and overexpressed IL-related cytokines. These monocytes emerged as the main peripheral drivers of the cytokine storm in critical patients. Alongside that, CD8 T cells showed exhaustion and heightened inflammatory activity, with IL-6 JAK STAT signaling switched on and the antiviral interferon alpha pathway suppressed. At the same time, the adaptive side of the immune system was weakened. Regulatory T cells and memory T cells were reduced, and several CD4 positive subsets showed dysfunctional or pro-inflammatory transcriptional states. Antigen-presenting CDC2 cells also declined and showed reduced MHC class II presentation capacity, further compromising antiviral control. Altogether, these changes map out a clear picture of immune dysregulation, where antiviral coordination breaks down and inflammatory programs dominate. The study also followed patients who received the recombinant WSK V102C vaccine. Notably, the high-dose vaccine dampened inflammatory monocyte activity and boosted NK cell and memory T cell compartments, suggesting that the vaccination can help recalibrate the immune system after severe dysregulation. So, overall, this paper provides a detailed single-cell view of how severe respiratory infection disrupts immune homeostasis and highlights potential therapeutic targets, especially around IL-6 signaling and the interferon alpha axis. Okay, so let's begin with a panel-by-panel discussion of the paper. If you're here more for a kind of broader look around the research methodology or any biases, data viz, the kind of paper craft of this, if you want to metagame things a little, then you're probably going to want to skip this panel-by-panel discussion, and you can do that using the chapters on YouTube. But if you're here for the actual paper, let's go. So in figure 1A, this is a nice study design overview. They show us that they have 17 patients across moderate, severe, critical, and convalescent groups. They generate almost 300,000 single cells for transcriptomic analysis. In figure 1B, the authors show a UMAP of all PBMCs clustered by major immune cell populations. Now, this gives us the global map of the immune landscape across these patients. Notice here how there's a clear separation of innate versus adaptive compartments, and this forms the foundation for later comparisons. Figure 1C shows how major immune populations shift with disease severity. Innate cells like CD14 positive monocytes increase, while adaptive cells like CD4 positive T cells decrease. Here, the take-home message is that severe disease equals more innate inflammation, less adaptive control. Figure 1E overlays cytokine scores onto the UMAP. Warmer colors in severe and critical cases show stronger inflammatory cytokine signatures. So here the take-home message is that cytokine activity escalates with disease severity. So in figure 2, the authors are trying to show us that CD14 positive monocytes drive the inflammatory storm. In figure 2A, they show that inflammatory scores across all cells. Notice that they're showing this clear increase in inflammation from moderate to critical disease. And apologies that the labels are hidden here, but we're going in increased severity from left to right. And you can see this increase shown in this area of the UMAP here. They're saying that inflammation tracks tightly with severity, and that's really one of the take-home messages across this whole paper. In figure 2B, CD14 positive monocytes stand out as the most inflamed population. This sets up the author's conclusion that these cells are major contributors to the cytokine storm. They're saying that CD14 positive monocytes are the primary inflammatory drivers. Then in panels C, D, E, and F, they are showing us pathway analyses and DEGs. So we're seeing cytokine production, IL-1 response, IL-6, JAK STAT activation, and upregulated inflammatory genes down here. These confirm this hyperinflammatory monocyte phenotype. They're showing that IL-6 and IL-1 pathways dominate. Monocytes become highly activated in severe disease. Moving on to figure 3, and here this is all about T-cell dysregulation. So parts A and B introduce the T-cell subsets identified through their single-cell sequencing approach. They're mapped here onto a UMAP and then a marker gene heat map as well. Multiple different T-cell states emerge, and this is a foundation for comparing severity here. In panel C, we're seeing the T-cell proportions as a Sankey plot. You can see how T-cell subsets shift with severity. Memory-like and regulatory populations decrease, while inflammatory and stress subsets expand. They're showing that severe disease shifts T cells towards inflammatory and exhausted states. Then panel D highlights exhaustion markers across T cells. So these increase markedly in severe and critical groups. Then the rest of the panels in this figure show pathway scoring for IL-6, JAK STAT3, and CD8 cells. And these pathways are strongly activated in severe disease. They're saying that IL-6 signaling drives dysfunctional T-cell activation here. Briefly, figure 4 explores CD4 T-cell phenotypes. Th17 cells become more inflammatory, while Tregs show stress-related and dysregulated transcriptional profiles in severe disease. The take-home message here is that severe infection disrupts regulatory balance within that CD4 compartment. Briefly, figure 5 shows that CDC2 cells exhibit reduced antigen presentation machinery, including downregulation of HLA class II genes. The take-home message here is that severe disease compromises antigen presentation, reducing adaptive activation. Now, figure 6 is where things start to get a little bit juicy. So we're looking at vaccination effects here, high dose versus low dose. In figure 6a, the authors show how the high-dose recombinant vaccine reduces monocytes and increases NK and CD4 T cells by day 14. The take-home message here is that vaccination shifts cell proportions towards a healthier balance. Nice. Then figure 6b confirms that inflammatory monocyte pathways, including cytokine signaling, are dampened after vaccination. So vaccine is calming that monocyte-driven inflammation. Also nice. In part C, these UMAPs clearly show decreasing inflammatory scores over time, particularly in the high-dose group. So vaccination progressively lowers inflammatory activation, and it seems to be dose-dependent. Then figure 6D finally shows NK cell upregulation of antigen presentation and regulatory genes after high-dose vaccination. So high-dose vaccination enhances NK cell regulatory and memory-related functions. Very nice. So finally, for this paper, figure 7 summarizes the mechanistic model. The authors are saying that IL-6 produced by monocytes signals into exhausted CD8 T cells, activating the JAK STAT pathways while interferon alpha responses weaken. Their take-home message is that IL-6 or STAT activation plus impaired interferon responses drive severe inflammation. Finally, just a little technical note from me. I am using the article in Press, as you can see from the watermark here, and it's not the final published version of the paper. In some places, the figure legends didn't quite match up with the order of the figures coming out. This happens. It's no big deal. I just hope that I got everything matched correctly. I think I did. But if you're an author on this paper or familiar with this work and you think I matched anything up wrong, please do let me know. And apologies if I did. So, if like me, you've not heard of this vaccine before, it's the one that's called WSK V102C. I wanted to give you a little overview of it if you're just interested in this vaccine compared to the other ones that are out on the market or the ones that you may have received yourself. So, it's a trivalent recombinant protein vaccine. So, that's XBB.1.5 plus BA.5 plus Delta. It's designed for immune-evasive Omicron lineages. So if you remember that Omicron variant of COVID-19 that came out a little while into the pandemic, it showed strong lab neutralization across these different variants, including JN1 and BA.2.87.1. It did offer moderate real-world protection in older adults, so we're looking at about 40 to 50% against symptomatic JN1 variant. However, it's mostly just been discussed in the scientific literature. So you'll see this in papers, not a huge amount of papers, but it definitely is out there. It's not really been mentioned in mainstream news as much, mostly just because of availability. I guess it's not in the Western world as much. Okay, so if you've been spoofing that you know what a UMAP is this whole time, then this slide is for you. So this is a UMAP. I've mentioned it throughout the panel by panel. And you've hopefully seen it if you've looked at any single cell papers before. I think it's vaguely intuitive, but it's worth going through it in a slightly more detail as well. So the UMAP is a 2D map of high-dimensional single-cell data. So we've got a lot more than 2D going on in this data, but we're trying to represent it in a way that it can be shown as 2D. So here, each dot of these many, many dots, like I say, about 300,000 dots in this case, is one cell. So 300,000 dots representing 300,000 cells. Their position reflects the similarity of the gene expression as seen in the single cell data. So clusters, these different colored areas, these form when cells share transcriptional profiles. The colors can then be interpreted to mean cell types or conditions. This means that the distance refers to their similarity, not anything to do with physical space. And the shape of the clusters isn't biologically literal. We're aiming to focus just on the patterns here. The key question when you're looking at one UMAP in comparison to another is which cell types expand, disappear, or shift in severe disease. I quite like figure 3, and I'm going to hone in on panels C and D briefly, just to give you a sense of some of the nice data viz that's going on here. So in part C, you can see a Sankey style plot. Now, this does a really nice job of showing how T cell subsets redistribute with disease severity. It's visually rich, but still easy to parse. And you can clearly see Tregs and memory-like CD4 subsets narrowing as the disease gets worse. It's a really strong visual link between the adaptive collapse and the severity gradient. I'm a fan. Moving on to part D, I think this is pretty nice as well. The authors have overlaid exhaustion markers directly onto these UMAPs, and it's extremely effective visually. This gradient-based coloring highlights exactly where the exhausted CD8 subsets sit, and you can see those hotspots grow in severe and critical cases, and it ties the mechanistic findings directly back to the cell map. Nice. Okay, so every paper has limitations, certainly no shade to the authors, but it's important for you as a reader to hone those skills around critical analysis of a paper. Not just if you're looking to talk about this paper in a journal club or critically assess it for an essay or exam, but also just to improve your own skills as a researcher or indeed as a scholar. So here's some of the key limitations that I noticed were a very small and unbalanced cohort. Now, this is certainly not unusual for a single cell paper. I think 17 patients or something like that. It's actually quite a lot. 300,000 cells, it sounds like a lot, but it's not. And in reality, if this was a different technique other than single cell, we would say that this is just simply not enough patients to draw any kind of meaningful clinical conclusions. Clinical trials have hundreds of patients for a reason. And if a power analysis was done in a kind of broader sense, I don't think this would be enough. We do sort of accept it in the single cell space that, as long as we've got a good handful of patients, that's good enough. And in some senses it is, but it is still a limitation. And hopefully once the technology becomes cheaper in the future, it's not something that we'll have to worry about as much. One major conceptual limitation here is that peripheral blood is not the same as lung tissue, and this is something that is kind of mentioned by the authors, but I don't know if it's really kind of come across strongly enough. Another is that the vaccination arm has a really low sample size, which, as I said before, is kind of a fault of the feasibility of the technique, but it's still definitely something to take into account. We're looking at a single time point analysis in acute infection. We do see some different time points when we look at the vaccine response, but really acute infection at one time point is something to pay attention to. There are some potential confounding from co-infections and persistent positivity. It is a bit of a heterogeneous cohort, so that's going to happen. And I suppose it almost feels cruel to the authors to say it as a limitation, but in a perfect world, we would really love to see some spatial biology here or functional validation. Everyone's going to have to be gazillionaires to do these kind of papers, but that is, of course, what gets it to a top-notch journal. And there's a reason for that. It helps us to understand the biology of what's going on in a really more of a big picture kind of way. I think spatial biology is going to be getting added to single cell papers more and more as we move forward. It's already happening a lot. And you're going to start to see people saying in practically every paper that it should have been added. So I really enjoyed reading this paper. I hope you did too. I think the authors did a pretty good job of showing their hypothesis that perhaps later in this severe infection, we get that kind of overactive innate immune system. Meanwhile, the adaptive immune system sort of weakens a bit. Of course, as with most single cell papers, we do have the caveat that it is a small sample size. It may be an awful lot of cells, but it's not very many patients. And so that, of course, is a caveat. It's one that you should probably take across other single cell papers that you're reading, because a lot of them really do have that same drawback. That being said, obviously with it being an extremely expensive technology, that's really the best that a lot of us researchers can do right now. And I think they've done a really, really nice job here of drawing logical conclusions and, kind of making it clear that this might not necessarily pan out across a big population, but it's certainly really interesting and persuasive out of the patients that they do have. If you're a student or a researcher, this is one that you should read a bit deeper if you're looking at things like cytokine storms, response to vaccinations in COVID-19, or dysregulated immune system between that adaptive and innate side, particularly if you're doing courses like infection and immune response. And then, moving on, if you want to have a look at some other deep dives on papers, please stick around and hit subscribe, as they say. I can only talk about open access papers here on YouTube in the sense that I quite like to show the PDFs and go through the figures, and I can't do that unless it's open access. But if you want to see what I'm reading this week in terms of, say, Nature, Cell Science, Lancet, NEJM, and just kind of tell you the main takeaways that I'm taking from the top tier research that's coming out, you can do that elsewhere on the internet. I'll drop some links in the description so you can have a look at that. But in the meantime, this is a really fun paper. I'm going to find more open access ones that we can discuss here in a similar manner. And happy reading.